All right, now that you understood the basic properties of enzyme kinetics in regards to the Vmax and in regards to the Km, now we need to represent these on two important graphic representations. The first one is what we call the Michaelis-Menten curve, or MM curve. And this is a hyperbolic representation of the relationship between the substrate and the enzyme. So on the x-axis right here, we have the substrate concentration in millimolars, and on the y-axis we have V, or the initial velocity of the reaction once the enzyme comes in contact with the substrate. And so how do you measure the initial velocity of the reaction is simply by knowing how much product are you producing per unit time, the rate of the reaction, right? So for example, the number 10 means that you're producing 10 products per unit time. And what we see over here is this, and if you're looking green, this curve is what we call what? We call it a saturation hyperbolic curve. All right, so that means that this curve is going to plateau. And we've seen this in multiple physiological states. For example, when it comes to the transport maximum, if you remember the Tmax of which substrate? Glucose. So for example, if we look at the proximal tubule, let's say this is the proximal tubule and we have a transporter, which is usually the sodium glucose transporter. This transporter can transport and reabsorb a certain amount of glucose, a certain concentration of glucose back to the blood vessels. And once it saturates, once that transporter can no longer hold any more glucose, then glucose will start, it'll start going into the urine. So this is what we call the transport maximum. So this transporter has reached the plateau. And it's the same exact thing when it comes to enzymes, and we just call it Vmax. It's the maximum velocity of the reaction. And so how do I know if I've reached the maximal velocity of the reaction? Well, we said the Vmax is governed by the number of enzymes. So if I have all my enzymes saturated and all my enzymes are present in the enzyme substrate complex, then I will reach maximal velocity of my reaction. Therefore, Vmax is not just going to tell you that this is the maximal velocity, it's also going to tell you that this is the point where all your enzymes are saturated. So if you increase the number of enzymes, you can increase the Vmax. If you decrease the number of enzymes, you can decrease the Vmax. And so I ask you, if I increase the number of enzymes, am I shifting the curve to the right or to the left? Exactly, neither. I'm not switching this curve to the right or to the left. What I am doing is I'm changing the Vmax, i.e. the plateau, and I'm shifting it upwards or downwards. So the next property is Km. How am I supposed to know how to represent the Km over here? Well, if we look right here, we said that the Vmax is 20. Half of the Vmax is going to be 10. So this point right here is what we call half Vmax. And the substrate concentration at half of Vmax is what we call the Km. And this is going to govern the affinity. Let's say in this graph, the Km is equal to 5 millimolars. So that means I need 5 millimolars of this substrate to achieve half of maximal velocity of this reaction. Let's say we shift this curve, let's say to the right or to the left. If we shift this curve to the right, then the concentration required, let's say, is 10 millimolars. So I need more substrate in order to attain the same velocity of the reaction. That means that the affinity has decreased. So as you can see, increasing the Km will decrease the affinity. And vice versa. If I shift this curve to the left, that means the substrate concentration that I require, let's say, is 1 millimolar. So I need just 1 millimolar of the substrate in order to achieve half of Vmax. So I need very little concentration, therefore the affinity of the enzyme to the substrate is very high. So this is how we have an inverse relationship between the Km and the affinity. Okay, so now let's see on the Michaelis-Menten curve, what happens if we change the Vmax or if we change the Km, and by what mechanisms can we do that? So let's say I want to increase the Vmax. That means I want to increase the amount of product produced per time. I want to increase the maximal velocity of the reaction. So we said that in order to do that, you need to increase the number of enzymes. But the thing is, there are various mechanisms to do that. So enzymes are proteins. They're made up of amino acids, and they need to be synthesized in cells. And therefore, what we can do is we can do something called induction. Now, there's going to be a lot of terminology in this slide right here, so I want you guys to focus. The term induction means an increase in gene expression. So we're going at the level of the DNA, intracellular at the nucleus, and we're increasing the gene expression for this specific enzyme so this enzyme can be synthesized more. This is different than what we call upregulation. Upregulation is another way we can increase the Vmax. Now let me give you the difference between them. When we say induction, we mean increase in gene expression, means we're increasing synthesis. Examples include, for example, insulin inducing lipoprotein lipase. These are different things that we're going to talk about later on. But it comes at the level of the DNA. We need to synthesize. Let me give you an example of upregulation. We remember that if you look at the endothelial surface, under the endothelial surface, we have what we call these little bodies called Weber-Palade bodies. And we know that these Weber-Palade bodies, they contain pre-synthesized and ready-made von Willebrand factor and factor VIII. These are factors that are required for the formation of a thrombus. And so whenever anything initiates thrombosis, if you remember from physiology, the Virchow's triad, anything that initiates thrombosis and initiates coagulation will upregulate these Weber-Palade bodies so that they can release preformed von Willebrand factor and preformed factor VIII. We're not synthesizing. They're already made. Let me give you another example. Insulin upregulates the GLUT transporter, which is what we call GLUT4, present in the adipose tissue and the skeletal muscle. It doesn't synthesize GLUT4. The GLUT4 is already pre-synthesized and it's ready in these vesicles intracellularly, and all you need to do is translocate it to the cell membrane. So this is what we call upregulation of an enzyme or upregulation of a transporter, a cell transporter. Usually, upregulation means that we're translocating a transporter from the intracellular environment to the actual cell membrane. So you have to understand the difference between induction and upregulation. Well, let's look at the opposite. What if I want to decrease the Vmax? That means I need to decrease the number of enzymes. So if we look at it, we can say the opposite of induction is the term what? Repression. Repression or silencing. And they're very similar terms, and what they mean is you're going to decrease the gene expression. And if you're good, you can remind me the term silencing. When it comes to DNA silencing, what is the process or the method that silences the DNA? Methylation, exactly. The other thing you can do is you can downregulate, which is the opposite of upregulation. And there's actually a third thing. It's what we call irreversible inhibition. And irreversible inhibition can be a multitude of things. It can be what we call non-competitive inhibition, which is usually what we call allosteric, or it can be competitive but irreversible. Let's explain these further. So let's see what non-competitive allosteric inhibition does. Let's say this is the inhibitor right here. What it does, it binds to another site, let's say right here, and it renders this enzyme inactive. So non-competitive allosteric inhibition means that this inhibitor right here will bind to another site, not the active site, and will cause an irreversible inhibition of this enzyme. Therefore, this enzyme cannot bind to the substrate. So if I had 3 enzymes, then I would be only left with 2 enzymes, and that means we're decreasing the number of enzymes. And if we're decreasing the number of enzymes that means we're changing the Vmax. Now, there are competitive inhibitors that are irreversible in nature. And if they are, even though they bind to the active site, they are not actually reversible. And therefore, that enzyme is going to be useless. Now, let's take a look at what happens if I want to change the Km. We said that the Km is the concentration of the substrate at half of Vmax. So if I want to shift this curve to the right, what am I doing? I'm increasing the Km, meaning that I'm decreasing the affinity. So the concentration of the substrate required to achieve half of Vmax is going to increase. And there are a multitude of examples for this. So the first of is actually competitive reversible inhibition. And if you think about competitive inhibition, you have the enzyme and you have an antagonist binding to the active site of the enzyme. Now, if it's a reversible inhibition, then all I need to do is I need to increase the concentration of the substrate in order to achieve the same reaction. That means in order to reverse this inhibition, you need to increase the concentration of the substrate, and therefore the curve will shift to the right. Another example is what we call desensitization. And desensitization means that this enzyme, even though it's present, its affinity has decreased. You can even put it in pharmacological terms. The potency has decreased. And therefore, you require more substrate in order to achieve the same effect. And the same thing if we shift the curve to the left. If I shift the curve to the left, I'm decreasing the Km, I'm increasing the affinity. And this is what we can call this what we call activation of an enzyme. If somebody says sensitization, which is the opposite, even the term potentiation of an enzyme, sensitization of a receptor. These are all terms used to tell us about changes in Km and changes in affinity. This is done by a bunch of processes called phosphorylation-dephosphorylation reactions. We're changing the shape of the enzyme in order to change its affinity and change its Km. The thing is, there is no clear-cut rule in the body that tells us, well, if you phosphorylate any enzyme, it'll activate it, or if you dephosphorylate any enzyme, it'll inactivate it. It depends on the enzyme. Some enzymes are activated by phosphorylation and inactivated by dephosphorylation, and vice versa. Some enzymes are inactivated by phosphorylation and activated by dephosphorylation. It all depends on the enzyme. Now that you've understood the graphic representation of the Michaelis-Menten curve, let's move on to the Lineweaver-Burk plot. Now, Lineweaver and Burk, what they did is that they said, okay, we have a Michaelis-Menten curve that is a hyperbolic representation of enzyme kinetics. But scientists, they look to optimize experimental studies and make them as easy as possible. So what Lineweaver and Burk did, they said, we're going to transform this Michaelis-Menten. It's going to be the same thing, but instead of it being a hyperbolic plot, we're going to try to make it a linear plot. You can see right here that the control over here is a straight line. And from mathematics, how many points do you require to draw a straight line? 2. Exactly. You only need 2 points. Therefore, in their experimental studies, you just need 2 experimental measurables, and you can go ahead and represent the kinetic analysis of an enzyme or a receptor or a transporter at any given concentration of the substrate. So that's why Lineweaver and Burk, they actually made it much more easier. Of course, you might be thinking, it's not easier for us because we have to just sit there and deal with this very difficult looking curve. But let's take it and analyze it one by one. So what they essentially did is they did what we call a double reciprocal plot. So they switched up things. So instead of on the y-axis having a Vmax, we have a 1 over Vmax. Instead of on the x-axis having the concentration of the substrate, we have 1 over the concentration of the substrate. And in fact, the other thing that they did is that in order to represent the Km, they had to extend the x-axis. And instead of it becoming 1 over Km, it's going to have to become negative 1 over Km. See, the whole idea about the Lineweaver-Burk plot is that we're not really looking at this slope. If you look at this slope, we don't really care about this slope. The slope, in fact, if you want to calculate what this slope represents, it actually represents the Km over the Vmax. But for us, we don't really care about that. What we care about is the intercepts. Actually, when you're looking at this curve, really looking at the slope itself is going to confuse you. you want to look at the intercepts. What do we mean by the intercepts? The point at which this slope crosses the y-axis and the point at which it crosses the x-axis. So the y-intercept represents 1 over Vmax. Wherever the slope crosses the y-axis, that's 1 over Vmax. And wherever the slope crosses the x-axis, that is negative 1 over Km. Negative 1 over Km, because it's crossing on the opposite side of the zero. So now that we know these intercepts, let's see what happens when we change the crossing of the slope. So if we look at this slope, let's say this slope crosses at a point at the y-axis that is higher than the control over here. Let's say it comes over here and crosses right here. Sorry for not having a straight line. Let's say it crosses a higher point. So now 1 over Vmax is increasing, right? It was like, let's say 0.3, now it's like 0.4. So you have an increase in the 1 over Vmax. What does that mean mathematically? Well, that means that there's a decrease in the Vmax. Therefore, if your slope is shifting right here, what are you doing to the enzyme? What are the 3 things that we discussed before? Exactly. You're either repressing or silencing, you're down-regulating, or you're non-competitively inhibiting. The slope has sort of shifted higher, so it's crossing at the y-intercept at a higher point. However, because this is a reciprocal, everything is going to switch. And the same thing over here. If I'm getting a slope right here, and it's crossing at a lower point, that means, if it's crossing at a lower point, let's say right here, that means we're doing the opposite. So the 1 over Vmax is decreasing, and therefore the Vmax is increasing. And this would be represented as a change in the enzyme as what? This represented as induction and upregulation. These are the parameters that we talked about. Induction, which is an increase in the gene which is an increase in the gene expression, upregulation, which we talked about. Let's look at the Km. The Km is a little bit trickier. Why? Because not only are you reciprocating once, you're actually reciprocating twice for the Km. Because not only are you going 1 over Km, but you're also adding a negative. And therefore, any shift, let's say negative 1 over Km shifting over here towards the right. So if it's shifting towards the right, that means there's an increase in the negative 1 over Km. All right, focus with me. This is math. Increase in negative 1 over Km, meaning it's becoming more positive. That's why it's increasing. Because you're reciprocating twice, even though this might be like, oh, this is supposed to be reciprocal, that means there would be a decrease in the Km. No. You're reciprocating twice, and therefore there's actually an increase in the Km as you shift towards the right. And vice versa. If I shift towards the left, there's a decrease in the negative 1 over Km, and therefore there would be a decrease in the Km because of this double reciprocal action that we're having. If we go ahead and represent this, an increase in the Km means a decrease in the affinity, which means we're talking about what? We're talking about inactivation, right? We're talking about desensitization of a receptor or an enzyme. We're talking about competitive inhibition, all right? Many different drugs that are competitive antagonists, okay? So this is very important because they can bring you a drug which is a competitive inhibitor, and they can ask you to represent it on a Lineweaver-Burk plot. Remember, the change over here is in the x-axis. Nothing is changing in the y-axis. So if you go ahead and say there's competitive inhibition, and it's shifting towards the right, you can see right here, look, it's crossing at the same point at 1 over Vmax, because I'm not changing Vmax. However, it's crossing towards the right at the x-axis. And the same thing if it's shifting towards the left, but this time we're going to talk about what? We're going to talk about activation. We're going to be talking about potentiation, and sensitization. So these are the ways that we look at the Lineweaver-Burk plot. Remember how to represent the Km and the Vmax on the Michaelis-Menten and the Lineweaver-Burk plots, remember what are the mechanisms by which you can change the Vmax and the Km on the Lineweaver-Burk and the Michaelis-Menten plots, and remember the different actions. What does induction do? What does activation do? What does competitive and non-competitive inhibition do? Because they can bring you a multitude of different questions and just give you these two graphs. So if you understand the concept, you're ready to answer any of the questions in regards to enzyme kinetics. Thank you.